I think I'll just go ahead and ask Dr. Bikini to introduce the HPV session.
All right, well, good afternoon, everyone. It's with some trepidation that I come to the, but there is no vote scheduled for this HPV work group vaccines session. So we'll go forward. So today's session, we're going to continue to provide clinical trial data from the investigational 9-valent HPV vaccine, and then discuss the policy questions that have been developed by the work group and the approach to grade. And then we're going to have a discussion about current information on 2-dose HPV vaccine schedules. Then to give you an update on vaccine workgroup activities, we are working to prepare for the expected licensure of the 9-valent HPV vaccine in late 2014. And we're continuing to review the data on 2-dose schedules. We've had a GSK presentation on their 2-dose development program in 2013. And recently, the work group did review data based on a presentation on the 2-dose schedule data that's available. Here's the timeline that the work group has for consideration of the 9-valent HPV vaccine. As you know, in October, at the October meeting, we presented an overview of the 9-valent clinical vaccine clinical program. And in February of this year, had a presentation on attribution of types in HPV-associated disease, and some initial clinical trial data being presented by Merck. Today, we'll have additional clinical trial data presented by Merck, and as I mentioned, the grade policy questions. In October, we expect to present information on health economics, present the grade analysis, and then provide some recommendation options for discussion. And then in February of 2015 is when we expect the earliest possible need to have a vote. So this is the formal outline of today's session. Dr. Elaine Luxemburg from Merck will provide additional trial data from the 9-valent HPV vaccine clinical trials. Then Dr. Eileen Dunn will follow with policy considerations and approach to grade for the 9-valent vaccine. And then Dr. Lori Markowitz will present the information on 2-dose schedules for the bivalent and quadrivalent vaccine. So these are the workgroup members. I want to thank them all. We have a number of very vigorous phone calls with a lot of discussion and a lot of input from each of the members, and I appreciate all the input that we've received, and I especially want to highlight Lori Markowitz as the CDC lead, who has kept a good organization for the group and kept us moving forward. So with that, we'll turn this over to Dr. Luxemburg to begin the initial presentation.
Okay, good afternoon, everyone. This is going to be the second part of the presentation of clinical data from the 9-valent HPV vaccine program. You have a few abbreviations that you can consult on your handout. And last time we talked about the 3 pivotal studies in the program. Protocol 1, the efficacy study in young women. Protocol 2 and 9, immunobridging studies in adolescents. And so we talked about the data at length. These studies are completed, continuing as longer-term effectiveness and immunogenicity studies. Today, we'll talk about the 3 other studies that have been completed, and they are more supportive studies. So we have 2 concomitant use studies, and 1 study unique to the program to assess 9-valent vaccine in prior Gardasil recipients. And just to mention that we have also other phase 3 studies ongoing. We have another study in young men, which is an immunobridging from the core efficacy study of young women to men. And then we have also a 2-dose, so alternative regimen study as well, and also an immunobridging study. So just as a summary of what we talked about last time in February, we found out that all efficacy and immunogenicity objectives were met in the pivotal study, and especially we demonstrated non-inferior immunogenicity with respect to the 4 original types that are common with the quadrivalent vaccine. Approximately 97% protection against disease due to the additional types, and the efficacy was extended to the pediatric population based on non-inferior immunogenicity. The adverse event profile was similar to that of the 4-valent, except that we found more injection site adverse events, mostly mild to moderate in intensity. And today we'll talk about the 2 concomitant use study, Protocol 5 and 7. We'll talk about Protocol 6, evaluation in prior Gaza recipient, and finally, integrated summary of safety, including all 6 studies. Protocol 5 is to assess co-administration of the 9-valent with 2 vaccines that are commonly used in adolescents, Menactra and Adacel. Menactra is a meningococcal vaccine, Adacel a Tdap vaccine commonly used in this country. And we all know what a concomitant use study is, so that's just a brief summary of the design. At day 1 in the concomitant cohort, subject received 9-valent concomitantly with Menactra and Adacel, or 9-valent alone in the non-concomitant group. The non-concomitant group receives Adacel 1 month later. And then all subjects receive at month 2 and month 6 their second and third dose of 9-valent. And these are the data. So with respect to the 9-valent HPV vaccine antigens, you see the concomitant cohort in blue, greenish blue, and the non-concomitant cohort in orange. And it's pretty obvious that the immunogenicity is the same. You have the GMT ratio at the bottom, and the non-inferiority criterion was met. Now looking for all 9 types. Now looking at seroconversion rates, you can see that it's hard to beat these. They are all the same. And then looking now at the concomitant vaccine antigens, so let's look first at Menactra. Menactra contains 4 Neisseria meningitidis serogroups, A, C, W, 1, 3, 5, and Y. For all 4 serogroups, non-inferiority criterion was met as well, which means we have really no substantial difference between the concomitant and non-concomitant cohort. Looking now at the Adacel antigens, we are looking here at diphtheria and tetanus. We are looking at serostatus cutoff of 0.1 international unit per mil, and then also a more stringent cutoff of 1 unit per mil. And for both antigens, the non-inferiority criterion is met as well. We have a very similar immunogenicity in both cohorts. Looking now at pertussis antigen, we have 4 antigen. And the abbreviation are in your list of abbreviations. So briefly, we met also non-inferiority of GMT for all 4 antigens. So conclusions are simple. It's, the co-administration is possible. There is no interference in terms of antibody response. We didn't show here safety data, but the safety was unremarkable. The vaccine was well tolerated. Protocol 7 is another concomitant use study. It's concomitant use with Repevax. It's another Tdap vaccine, and that vaccine contains also inactivated poliovirus. It's used in Europe, so it's not directly relevant to this country, but to be complete, we just provide you a summary of the findings. So it's very consistent with Protocol 5. There is no interference in terms of antibody response with any of the vaccine antigens, and also no safety concern. Okay, let's go now to Protocol 6. Protocol 6 is an evaluation in prior Gardasil recipient. So I'll spend a little more time because it's a unique study to this program. First point is that this is primarily a safety study. And what we had in mind here is help a healthcare professional, when the 9-valent, should the 9-valent be licensed and recommended, when individual would come to their primary care physician for advice, there is some data because there may be individual who had been vaccinated before looking for additional coverage, people with unknown history of HPV vaccination. I mean, there are a number of scenarios. And so the goal is primarily to show that it is safe, or to document that it's safe to administer the 9-valent when you have received the 4-valent. We obviously looked also at immunogenicity. So subjects were randomized to 9-valent vaccine or saline placebo. Once again, it's a safety study, so that allows the safety profile of the vaccine to be more completely defined. And we collected the usual endpoint for immunogenicity and safety. Study was conducted in girls and young women 12 to 26 years of age, because it's the most likely age for people to look for a follow-up vaccination. And this is the study design, again, more as a graphic. Subjects received 3 doses of commercial Gardasil before entering the study, and it had to be documented by medical record, and then entered the study. There was a time interval requested of 12 months between the last dose of Gardasil and the first dose of whatever they receive in the trial. And the reason is that we didn't want a subject at their peak of immunogenicity immediately after the 3rd dose. Wanted a population of subjects that was homogeneous in terms of antibody titer. So 12 months was a reasonable time to wait. These are safety data. We show safety first because it was the primary objective. You can see 9-valent versus saline placebo. It's pretty clear that the vaccine is well tolerated. There are very few discontinuation due to vaccine-related adverse events, and very few vaccine-related AE as well. We are looking really at the difference between 9-valent and placebo in terms of injection site, not a surprise. And that was really important to look at because, this subject in the 9-valent cohort will have received 6 doses of HPV vaccine in their lives. So, is it safe, especially in terms of reactogenicity? So let's look further in injection site. And, you can see that the frequency of injection sites are, if you remember, consistent with what we've seen in Protocol 1 last time, with pain, swelling, and erythema being by far the most common. And here again, most injection site adverse events were mild or moderate in intensity. So the tolerability appeared acceptable. Let's look now at immunogenicity. The primary endpoint was to look at, the secondary immunogenicity objective was to look at seroconversion rates. And you can see that both in the girl subset and in the young women subset, the seroconversion rates are high, which means the vaccine is very immunogenic in both populations. Looking at GMT. So you want to look first at the 4 original types, type 6, 11, 16, and 18. And what you can see is that at day 1, there is already a substantial GMT. Not a surprise, they are all prior Gardasil recipients. After 1 dose at month 2, we have a substantial increase. And after 2 more doses, there is not much of a change, which means you get most of the increase after the first dose. It's not a surprise, it is a memory response, and it's very clear here. Looking now at the additional types, the picture is different. In general, there is no or very little antibody at day 1. And then there is a substantial increase after the first dose, and there is further increase after the 3rd dose, with some of the type where, we have more than half a log difference. So So for these, there is a benefit in having more than one dose for the additional types. This is in the girls. We show the girls and the young women separately because immunogenicity is age dependent. So looking in young women, I'm not going to spend as much time, but the profile is essentially the same. Memory response for the original types, for the new types, more a pattern that looks like a primary response. So summary for Protocol 6. We have an acceptable safety profile, and the vaccine is highly immunogenic. Things that are important, a question that comes very often. This study investigated 3 doses of Gardasil followed by 3 doses of 9-valent. It didn't investigate mixed regimen, partial dose, all of this. That was not the goal. The goal was, as we mentioned, to provide information for healthcare professionals. And also it was primarily a safety study, so we didn't do an immunogenicity comparison, let's say in subject who received 3 doses of 4-valent followed by 9 doses of 4-valent compared to subject who received 9-valent alone. There was not a 9-valent alone cohort here. Obviously, we are interested in that comparison, but it has to be a cross-study comparison. And so we did that exploratory analysis comparing immunogenicity after 3 doses in Protocol 6 in prior Gardasil recipient versus what happens in all the other studies in the program. And so the goal was to compare immunogenicity. It's cross-study. Why did we do that? It's because it's known that previous antigenic encounter can affect the immune response of a related antigen. And what we have to keep in mind is that all these HPV types, we use the L1 protein. The L1 protein has a very high, there is a very high homology between types. And we give here an example, between type 16 and type 33. Type 16 and type 31, there is approximately 83% homology, which means that you cannot say that subjects who are prior Gardasil recipients are strictly speaking totally naive to the additional type. So it's of interest to look at what's going on. We remember the original antigenic scene, in the flu vaccine where there is, a response can be affected. So let's look at the data. First, let's look at seroconversion rates. So you see 9-valent vaccine after 4-valent. This is Protocol 6. And you see 9-valent vaccine only. These are all the subjects in the program. And the two groups are not distinguishable in terms of seroconversion rates. Very, very immunogenic in both groups. Now let's look at GMT. So these are the GMT in young women. In blue, we have the cohort receiving 9-valent after 4-valent. In orange, we see the cohort receiving 9-valent only. And in yellow is a natural HPV infection. So let's focus first on the original types. And here what we see is that the GMT in subject who received 9-valent after 4-valent is higher than what we've seen in the combined other studies where subjects received 9-valent alone. This is consistent with memory response once again. Looking now at the new types, here we see that the picture is reversed and the immunogenicity is lower with respect to the new types, and you have the GMT ratio at the bottom, which is consistent with what I tried to introduce, original antigenic scene. And so the GMT in the 9-valent after 4-valent group is lower than 9-valent alone, but still higher than with natural HPV infection. So this is a comparison with young women, Now if we look at girls who received 9-valent after 4-valent compared to women who received 9-valent only, so women who received 9-valent only, the efficacy population, then the difference is less in respect to the additional types. So for some types, the GMT are comparable. For some other, it's slightly lower. And for the original types, GMTs are higher. So in summary, the first thing this is a cross-study comparison, so results need to be interpreted with caution. This is exploratory. But overall, it's consistent with prior observation that if you are exposed to an antigen and then following that with an antigenic variant, there is a chance you have a lower immunogenicity. Practical consequences are probably limited. We've demonstrated that the seroconversion rates are high in all subjects. Even though we have difference of GMT between the groups, the significance of the difference is unknown because there is no known threshold of antibody protection. Also in Protocol 1, which is a pivotal efficacy study, we've shown that antibody titers with respect to the additional types are not correlated with lower efficacy. And once again, that brings to mind the original antigenic scene with flu, and in that field, it's been demonstrated that revaccination is associated with higher protection. So the impact is pretty limited also for that other vaccine. Looking now at the last part of our presentation, integrated safety data. So now you've seen data or you've been informed of the result of 6 studies. Let's look at the integrated safety for all 6 studies. And before getting there, let's just as a reminder, what you've been shown last time is comparing the 9-valent and the 4-valent. The profile was comparable. Safety profile was comparable. Only a higher frequency of injection site adverse event, as I mentioned, mostly mild to moderate in intensity. So that's what you've seen last time. In approximately 7,000 subjects in each cohort. Now looking at the integrated database, so now we're looking at over 13,000 subjects. Safety profile is comparable to what we've seen so far in Protocol 1. Vaccine well tolerated. Most of the adverse events are injection sites, and there are very few, serious events that are vaccine related. Looking at injection sites, pain, swelling, and erythema are the most common, and the frequencies are consistent with what we've seen previously in Protocol 1. And here again, for the integrated database, most injection site adverse events are mild to moderate in intensity. Vaccine-related systemic adverse event with an incidence of more than 1%. It's again a short list. A list of events that are everyday life events and common in any vaccine trial. We had 5 vaccine-related serious adverse events. Just only 2 of them required hospitalization. They all resolved, and nothing that's unusual or that has not been seen before in the 4-valent program. So in terms of safety, we have an adverse event profile that's favorable in over 13,000 subjects, few discontinuation, few vaccine-related serious adverse event, no vaccine-related death. And the adverse experience profile is generally comparable to that of the 4-valent. We've seen also that the 9-valent vaccine is well tolerated in prior Gardasil recipients. Additional analyses were done to look at subgroup by age, gender, race, ethnicity, and we didn't see much difference either. So overall summary of part 1 and part 2, we've demonstrated non-inferior response with respect to the original types compared to the 4-valent vaccine. High efficacy against disease associated with the new types. Efficacy findings in young women could be extended by immunogenicity in boys and girls. Vaccine was generally well tolerated. Adverse events profile similar to that of the 4-valent vaccine. 9-valent vaccine can be co-administered with Menactra and Adacel. It is generally well tolerated and highly immunogenic in prior 4-valent recipients. And as a reminder, this is still an investigational product under review by the FDA. Thank you very much.
Thank you. Should we move to the next and cover the 9-valent questions altogether? Is that? So Dr. Dunn.
So good afternoon. In this presentation, we're setting the stage for grade review of 9-valent HPV vaccine in October of this year. In order to frame the policy questions, I'd like to review again the attribution of HPV types to cervical precancers and cancers. And this was presented to ACIP in February of this year. I'll then discuss 9-valent HPV vaccine policy questions for consideration, as well as the available 9-valent HPV vaccine studies, the conclusions from the work group on critical outcomes, the PICO for the primary policy questions, cost effectiveness considerations, and our next steps. So this is the HPV type attribution by cancer site in the US, and in red is the attribution to HPV type 16 and 18, and yellow is the additional 5 types that are prevented by 9-valent HPV vaccine, and green are other HPV types, and gray are HPV negative. And as you can see, HPV 16-18 is attributed to most cancers. The 5 additional types are attributed to between 14 and 18% of cervical, vulvar, and vaginal cancers, and 6 to 9% of anal, penile, and oropharyngeal cancers. So this is an overview of HPV 16 and 18, as well as 5 type attribution to CIN2+ and HPV-associated invasive cancers. About 50% of CIN2 are attributable to HPV 16-18 and about 25% to the 5 additional types. As far as HPV-associated invasive cancers, 62% are attributed to HPV 16-18, and this is similar for females and males. 11%, an additional 11% of cancers are attributed to the 5 additional types in 9-valent HPV vaccine, and this is different for females and males, with 14% in females and 5% in males. I'd like to summarize the 9-valent HPV vaccine policy questions for grade. The primary questions are, should 9-valent HPV vaccine be recommended routinely for 11 or 12-year-olds? And should 9-valent HPV vaccine be recommended for females aged 13 through 26 years and males aged 13 through 21 years who have not initiated or completed the vaccine series? The ACIP HPV workgroup was interested in considering these policy questions for males and females at the same time for programmatic reasons. Although because the outcomes are different in females and males, as well as the attributable fraction due to 9 types, the PICO would be considered separately for males and females. The workgroup would like to consider a recommendation for adult males with available data in October of this year, even though there would not be an adult male indication for 9-valent HPV vaccine at the time of first licensure. Other policy questions included, should girls and young women who received 3 doses of quadrivalent HPV vaccine be revaccinated with 9-valent HPV vaccine? And should 9-valent HPV vaccine be recommended for men who have sex with men through age 26 years? So you've seen this slide before, which summarizes 9-valent HPV vaccine studies for the initial filing. Studies include the efficacy in adult females, which was presented earlier to the ACIP and earlier this year. There's also immunogenicity studies in boys and girls, as well as young women. The concomitant use studies, as well as the study that was highlighted earlier today, which is the safety study of 9-valent HPV vaccine in prior HPV-4 or quadrivalent vaccine recipients. An additional study will likely be available in October, as I highlighted earlier, of immunogenicity in adult males. So I would like to highlight some features of the data available for the policy questions. As a reminder, The main trial was a comparison of quadrivalent vaccine and 9-valent HPV vaccine. There was no placebo arm. For adult females, there'll be efficacy and immunogenicity data. In adult males, there'll be immunogenicity data. As far as the data on revaccination with 9-valent HPV vaccine in females, that's data is available in females only. And the immunization schedule in the trials was 0, 2, and 6 months. In order to address the critical outcomes to consider for grade, we surveyed the ACIP HPV work group in May of this year on key outcomes. This includes scoring outcomes in females and males and benefits and harms. We received 21 responses, and the following slides summarize the opinions on critical outcomes. We also surveyed AIM members and received 26 responses, and the feedback was consistent with that of the ACIP HPV workgroup. And this table lists the critical outcomes for females. These include cervical cancer, cervical precancer, anal cancer, vaginal vulvar cancer, CIN, definitive therapies, and oropharyngeal cancer, as well as serious adverse events and anaphylaxis. As a reminder, for many of these outcomes, there is no direct data from the clinical trials. And so in this table, we've listed the outcomes in which there's direct data, including cervical precancer, CIN, definitive therapies, SAE, and anaphylaxis. For some outcomes, there'll not be direct data, but there is indirect data, such as data on cervical precancer, immunogenicity, and vaginal and vulvar precancers. For males, the critical outcomes included anal cancer, oropharyngeal cancer, SAE, and anaphylaxis. And as you can see from this table, there'll be direct data for SAE and anaphylaxis only, and indirect data will include immunogenicity. So for the next few slides, I'll summarize the PICO for the primary policy questions, or the population intervention comparison and outcomes. For the question of routine vaccination at 11 or 12 years, the population is females immunized at age 11 or 12. Intervention is 9-valent HPV vaccine, and comparison is quadrivalent vaccine. And the critical outcomes were highlighted earlier. The primary objective is an evaluation of HPV 6, 11, 16, and 18 equivalence, or non-inferiority, and evaluation of HPV 31, 33, 45, 52, 58 superiority. For males, the population is males immunized at 11 or 12 with a similar intervention and comparison, and the critical outcomes, again, were highlighted earlier. The primary objective for the male evaluation will be evaluation of 6, 11, 16, 18 equivalents. We did not elect to evaluate superiority of 5-type HPV outcomes in males, as the attribution of 5 types, these 5 HPV types, to male critical outcomes, such as anal cancer and oropharyngeal cancer, is limited. For the PICO for the question in older females and males includes the population of females immunized at age 13 through 26 with the intervention of 9-valent HPV vaccine in comparison to quadrivalent vaccine. And again, critical outcomes were highlighted before. And again, the objective is the evaluation of HPV 6, 11, 16, 18 equivalents, as well as 5-type superiority. For males, this is slightly different. Males immunized at age 13 through 21 with the same intervention and comparison. Critical outcomes were highlighted before. The objective for males is evaluation, again, of HPV 6, 11, 16, 18 equivalents. And I have highlighted the data numerous times, but this is, again, the data evaluated that will be evaluated for those primary policy questions. That includes efficacy and immunogenicity in adult females, immunogenicity in boys and girls, and immunogenicity in adult males. And again, a reminder that the data for immunogenicity in adult males is likely to be available in October of 2014 for our great consideration, but will be pending FDA approval for an adult male indication at the time of primary licensure. So cost effectiveness is considered, as you know, for grade. For 9-valent HPV vaccine, we'll present cost effectiveness for males and females separately and combined in October of this year. Cost effectiveness of revaccination with 9-valent HPV vaccine after receipt of 3 doses of quadrivalent vaccine will be presented as well. A variety of models will be summarized, both CDC and external models that use US data. In summary, we're hopefully provided the foundation for our next presentation to ACIP on grade for 9-valent HPV vaccine considerations. So the policy questions in PICO have been developed, and as far as next steps are concerned, we'll develop tables of evidence on 9-valent HPV vaccine and present to the work group. And we'll also prepare for presentation on cost effectiveness and grade to the ACIP in October of 2014 for possible recommendations in early 2015. So thank you.
Let's see if there's any questions or comments on the last two. Yes, Dr. Karen.
So yes, I have one question for Dr. Dunn and one for Dr. Luxenberg. So for you, Dr. Dunn, the question was, I know in a previous presentation you had spoken about ethnic and racial disparities in terms of the 5-valent, and I didn't know if it was for carriage, for CIN2, or for cancer, but can you say anything about that?
In our previous presentation in February, it was me, it was Dr. Mona Soraya and Dr. Harir. That's okay, we all look the same sometimes up here. So we presented attribution to 5 types and then the 9 types to CIN2 plus and cancers. And there were race ethnicity differences for the cervical precancers in particular. Primarily for the invasive cancers, the attribution of 16-18 is the same across race ethnicities.
And the question I had for Dr. Luxenberg is, when will there be data available on the 2-dose regimens for the 9-valent vaccine?
So we anticipate analysis to be conducted before the end of next year.
Thank you.
Dr. Schaffner? Okay. Dr. Harrison?
Yeah, so my question was a related question. So you showed the change in antibody titer between doses 1 and 3, but how about between doses 2 and 3?
It was not shown in this particular study. In protocol 1, we did post-dose 2 and post-dose 3, and there is an increase, but it's relatively small, probably around 30%, something like that.
Dr. Vasquez.
Yeah, similar question. When you showed immunogenicity data in the cross study, that's comparing what happened to individuals after 3 doses of the 9-valent. Do you have information on 1 dose or 2 doses?
We don't have that type of information for a cross-study comparison. In Protocol 6, we studied only post-dose 1, and in Protocol 1, we studied only post-dose 2.
Dr. Rengel.
Yeah, I was wondering if you could just, Dr. Dunn, elaborate a little bit on the decision not to look at the critical outcome in males for HPV 31, 33, 45, 52, and 58. And I'm sure the data are scanty, but I'm just curious if that really is not possible.
It absolutely is possible, but the focus of the work group was that the attribution, and maybe I should show the previous slide again of the attribution to these specific 5 types is less for males than for females. And certainly this isn't separating males and females, but you can see the attribution for penile is 9%. The overall attribution for males is 5%. We have not yet presented cost effectiveness data, and I think that will be presented in October of this year. and which brings up additional questions around cost effectiveness of 9-valent for males versus females.
Dr. Karen.
Sorry, I just wanted to ask Dr. Luxemburg a follow-up question. So I just wanted to understand, I know that there is a formal study looking at 2 doses, but are there existing data for naive individuals getting 2 doses of 9-valent vaccine? Perhaps maybe they received 3, but you have immunogenicity data after 2 doses. Do those data already exist, or do they not exist and you're anticipating getting them with the upcoming study?
The only data we have for 2 doses are in the context of Protocol 1, young women 16 to 26. I think it's not completely relevant to what the ACIP would be interested for 2 reasons. One, it's not the group where a 2-dose regimen would be considered, which would be more pediatric. And the other is that the schedule is different also from what would be considered, because subjects received at day 1, month 2, and month 6. So 2 dose, any subject who received 2 doses would be with 2-month interval. So it's also very different. So that long explanation to say, no, we do not have relevant data at this time.
Okay, let's move along to Dr. Markowitz.
Okay, well, the purpose of today's presentation is to inform and update the committee on the accumulating data on 2-dose HPV vaccination schedules. And what I'd like to do first is provide some background, then review some recommendations and regulatory approvals around the world. Then I will present the data on 2-dose schedules. This is not a comprehensive review, and it's certainly not an evidence review, but it's just providing an overview of the landscape of data available for the committee. Then I'll review information on countries using 2-dose schedules, considerations for the US, and the work group plans. As background, we have 2 licensed products in the US. I think everyone knows the quadrivalent and the bivalent. They both are virus-like particle vaccines using the L1 protein. The vaccines contain different adjuvants. And the license schedule for both vaccines is a 3-dose schedule, with the second dose given 1 or 2 months after the first, and the third dose 6 months after the first dose. For licensure of the currently available vaccines, large efficacy trials were performed in 15 to 26-year-old females, and the endpoints were precancer lesions. And then bridging immunogenicity studies were conducted in 9 to 15-year-olds, an age group in which efficacy trials were not feasible. And licensure in this age group was based on the non-inferior antibody response compared to the age group of women who had been in the efficacy trials. Now, over 99% of vaccinees developed antibody after vaccination, and the antibody titers in the younger age group was higher than those in the women in the efficacy trials. And this is figures from a paper published in 2007 showing GMTs for HPV 6, 11, 16, and 18 1 month after the third dose by age and enrollment from 9 to 23 years. And you can see for all of these types, the GMTs were highest in the youngest age group and decreased with increasing age at vaccination. And very similar findings by age are available for the bivalent vaccine for HPV 16 and 18. Now, the main basis of protection is felt to be neutralizing antibody, but the minimal protective antibody threshold is not known. The high efficacy found in the vaccine trials has precluded identification of a protective titer. But vaccination induces antibody titers that are much higher than after natural infection. In clinical trials of the quadrivalent vaccine, some vaccinees lost detectable antibody for HPV 18 by the competitive Luminex immunoassay, but there was no loss of protection against the endpoints in the trials. Now, I think everyone knows there's been global interest in simplified schedules for HPV vaccine. This would be more convenient for providers, parents, and vaccinees. And it's possible that it could also facilitate implementation by reducing logistical challenges in some settings and also decrease resource needs. And the high efficacy found in the original trials prompted investigation of reduced dose schedules. Before I go into the data, I want to mention a few more things as background, and one is what's considered the immunologic basis of the HPV vaccination schedule. The 3-dose schedule with a series at 0, 1 to 2, and 6 months can be considered a prime boost schedule, with the first 2 doses being the priming doses and the third being the boost. And a 2-dose schedule at 0, 6 months can be considered a prime boost, with the second prime having been dropped. Memory B cells require at least 4 to 6 months to mature and differentiate into high-affinity B cells, and the 6-month interval between the first and the last dose is felt to allow the last dose to efficiently reactivate memory B cells. WHO's Strategic Advisory Group of Experts, or SAGE, on immunization considered a 2-dose HPV vaccination schedule in February 2014. At that meeting, they recommended a 2-dose vaccination schedule for girls if vaccination is initiated prior to 15 years of age, with the minimal interval between 2 doses being 6 months, and the interval may be extended to 12 months if it facilitates administration. And a 3-dose schedule remains necessary if immunization is initiated after the 15th birthday. And a 3-dose schedule also remains recommended for immunocompromised individuals, including those known to be HIV infected. SAGE reviewed a variety of data on 2-dose schedules for this decision, and I will review some of this today. This included a systematic review of the literature and the EMA assessment report for Cervarix, and both of these are available on the WHO website. I want to mention that the manufacturers have submitted data on 2-dose schedules to regulatory authorities in a variety of countries. The bivalent vaccine has regulatory approval for use as a 2-dose schedule in Europe, in the EU, and 5 other countries, as well as countries in Africa, Latin America, and Asia. And the quadrivalent vaccine also has regulatory approval for use as a 2-dose schedule in the EU and a variety of other countries. What is the current status of regulatory approval in the US? For bivalent vaccine, there has been no submission to FDA. For the quadrivalent vaccine, there are no plans for submission because Merck is focusing on their 9-valent vaccine, as you heard today, which is currently, and there's no data on 2 doses included in the BLA under consideration by FDA, although a 2 versus 3-dose trial has been initiated, and I'll review that trial later in this presentation. Now, with that background, I'm going to review some of the data on 2-dose schedules for the bivalent quadrivalent vaccines. Most of the data are from immunogenicity studies. There are limited data on efficacy from post-hoc analyses, and there are now data from post-licensure effectiveness evaluations. So first, I'm going to review the immunogenicity data, and much of this was presented at the SAGE meeting. This slide shows immunogenicity studies directly comparing 2- and 3-dose schedules. For each study, the table includes the countries where it was performed, the vaccine that was used, the age groups, and the schedules, and then the last column is the longest follow-up. There are 3 studies of the bivalent vaccine and 2 studies of the quadrivalent vaccine. I'll review these in more detail later. The first study is a GSK-sponsored study. The study was a dose-ranging study, and other groups were included other than those I've indicated here. And there are 2 publications from this study, the most recent published last month, having a 48-month follow-up. The second study is also a GSK-sponsored study. It's considered by GSK their confirmatory study of 2 doses. These data have been presented at 2 international meetings in the last 8 months, and the data have also been submitted to regulatory agencies. And the third study is a study conducted by researchers in Mexico. There are 2 studies of the quadrivalent The first one here is, neither of these were industry-sponsored. The first one is the Dobson study conducted in Canada, and this is the main published study that we have on the quadrivalent vaccine. It has been included in Merck's submission for regulatory agencies. This last study was conducted in India. It was originally planned as a randomized trial of quadrivalent vaccine with over 20,000 girls 10 to 18 years of age, randomized, received 2 or 3 doses. The original design was going to evaluate SIN outcomes, but the study was not completed as planned due to being halted by the government. And at the present time, data are only available on a subset for immunogenicity and incident infection, and the randomized design has been compromised. 4 of these studies had a group that included the standard 3-dose schedule at 15 to 26 years of age approximately, which is the age group of the women in the efficacy trials. And this was the main comparison that was made, was between antibody in this group with the experimental 2-dose schedules in the young adolescents. 4 studies also were designed to allow a direct comparison between the 2-dose schedule at 0-6 months and the standard 3-dose schedule in the same age group. And 1 study of the bivalent vaccine compared 2 different 2-dose schedules, a 0-6 month and a shorter interval of 0-2 months. And this comparison was made in several age groups. The next few slides are going to be figures from the systematic review that was conducted for SAGE. And this particular slide shows differences in the proportions seroconverting or seropositive between girls who received 2 doses and the women who received 3 doses. And they're constructed similarly. Estimates on the right favor 2 doses and on the left favor 3 doses, and the dashed blue line is the non-inferiority margin. So 4 studies, 3 of the bivalent and 1 of the quadrivalent contributed to this analysis. How there are many more data points because 16 and 18 are shown separately, and those also data points from different follow-up periods, and they're also separated into high and low-income countries, so it's a little bit complicated to look at. So the top half of this slide shows HPV 16. You can see that for all comparisons, there was no difference in seroconversion or seropositivity between the girls who received 2 doses and the women who received 3. And for HPV 18, on the bottom of the slide, you can see that there was no difference, and in fact, for HPV 18 in the 1 study of the quadrivalent vaccine, seropositivity was actually higher at 24 and 36 months for girls who received 2 doses compared to women who received 3 doses, although this was not statistically significant. And this slide shows the geometric mean concentrations from the same studies I showed in the previous slide, comparing girls who received 2 doses and women who received 3 doses 1 month after their last dose. Again, 4 studies contributed to this, 3 of the bivalent and 1 of the quadrivalent. And as you can see on the top is shown HPV 16, and in all of these trials, non-inferiority criteria were met. And for most of them, they favor the 2-dose schedule in girls. And same for the HPV 18. The next 2 forest plots are comparisons of girls of the same age receiving either the 2-dose or the 3-dose schedule. And this slide shows differences in proportions seroconverting or seropositive. Only 2 studies contributed to this analysis, 1 of the bivalent and 1 of the quadrivalent. And for HPV 16, you can see there was no difference between the 2 and the 3-dose schedules. And for 18, in the 1 study, which was of the quadrivalent, seropositivity was higher at 24 and 36 months in the girls who received 3 doses compared to the girls who received 2, but the lower confidence interval included the non-inferiority margin. And this is the forest plot showing the differences in the geometric mean concentrations in girls of the same age who received 2 or 3 doses 1 month after the last dose. There are 3 studies in this comparison, 2 of the quadrivalent and 1 of the bivalent. And for HPV 16, the first trial is the bivalent vaccine. You can see the geometric mean concentration is somewhat lower, but not significantly. And for the quadrivalent vaccine, the estimates were not different between the 2 schedules 1 month after the last dose. And for HPV 18, for both the bivalent and the quadrivalent vaccines, The geometric mean concentrations after 2 doses were lower, but not inferior to 3 doses. And the last, on the below, on the graph, you see the study from India, where here 2 doses actually seemed to give geometric mean concentrations that were higher than the 3-dose schedule. And finally, this is the last forest plot I'm going to show. This shows a comparison between the schedules with 2 different intervals between the 2 doses. All of these data points are from the same study of the bivalent vaccine, but what's shown here are the different age groups that were compared. And you can see for all the age groups, and both for 16 and 18, schedules with a 0-6-month interval resulted in higher geometric mean concentrations than those with the 0-2-month interval between the 2 doses. Now, what I'd like to do is review separately some of the data for bivalent and quadrivalent vaccines, so you can see a little bit more of the data that's available for each vaccine. For the bivalent vaccine, this will include some of the data that were in the forest plots. They include immunogenicity data from study 48, which was the Canadian-Germany study, and 070, which was referred to as the multinational study. These are the main data submitted to regulatory agencies. I will also present data from 2 post hoc analyses of efficacy trials that provide data on 2-dose schedules. So just briefly, again, this was included in the forest plot of the SAGE review. This is Protocol 048, which was also called Canada-Germany in the SAGE review. This is a dose-ranging trial as well as a 2 versus 3 dose trial. And there was a 48-month follow-up that was just published last month, and this was following up just the licensed formulation in 2 groups. That is the most important groups for our question, which are 2 doses in 9 to 14-year-olds and 3 doses in 15 to 26-year-olds. And the findings here are that all subjects remain seropositive for 16 and 18 by ELISA at month 48. The geometric mean titers were non-inferior for the 2-dose group compared to the 3-dose. And I'm showing here one of the graphs from the paper, which shows the really identical antibody kinetics for both groups, where the height of the antibody peaks after the last dose, then declines and reaches a plateau. And you can see the lines are almost completely superimposed for these 2 groups. This was the design of the 070, which was, I said, considered the confirmatory trial, the larger 2 versus 3 dose trial. The design was slightly different. There are 2 different experimental groups in the 9 to 14-year-olds, a 0-6-month and a 0-12-month. And there was a concurrent group of 15 to 25-year-olds. And the data from the 0-12-month were just presented last month and were not included in the SAGE systematic review. These are the data from that study, and on the left in the blue bars, what's shown is are the geometric mean titers 1 month after the last dose in the 3 study groups. On the far left is the 15 to 25-year-old women who received 3 doses, then the 9 to 14-year-old girls who received 2, and the 9 to 14-year-old girls who received 12. And you can see there's really no difference in geometric mean titers between these 3 groups. In the orange bars show the longest follow-up to date, which is 6 months after the last dose, and the data here are only available for the women 15 to 25 and the 0-6-month group. Geometric mean titers were slightly lower, but not significantly, in the girls who received 2 doses, and non-inferiority criteria were met. And you can see that antibody titers remain substantially higher than those after natural infection. Efficacy data are available from 2 post hoc analyses. I think we've shown some of these data to ACIP before. It's important to note that these studies were not designed to evaluate number of doses, but were the RCTs to evaluate efficacy of 3 doses. Data on this first slide are from the post hoc evaluation of efficacy against persistent infection in a bivalent vaccine trial conducted in Costa Rica. Among women enrolled in this RCT, about 20% received less than 3 doses. In this post hoc analysis, the endpoint was incident persistent infection that lasted 10 months. The numbers are small, but it shows that there was efficacy observed for those who received 1, 2, or 3 doses, with an efficacy of 84% for those who received 2 doses, not different from those who received 3. This is the second post hoc analysis, unpublished. These data are from the pivotal randomized controlled trial conducted by the manufacturer Patricia. In this large trial of 15 to 25-year-old women, compliance with the 3-dose schedule is high. Only 5% received 2 doses. And shown here are the 2 and 3-dose efficacy against 6-month persistent infection with 16 and 18. And you can see the numbers are very small here. The efficacy was 100% for 2 doses, 93.7 for 3 doses. Now I'd like to go on to the quadrivalent vaccine and go in a little bit more detailed data that are available for that. As I mentioned, the main data available for 2 doses are the immunogenicity data from a study conducted in Canada in 2 doses in younger adolescents versus 3 in older women. I will also mention some data available from a randomized controlled trial of 3 different schedules, of different 3-dose schedules in girls 11 to 13 years old that provide some data on 2-dose schedules. Again, this study was also part of the SAGE systematic review, and I'm just reviewing them here in more detail. This is the design of the study. 9 to 13-year-old girls were randomized to receive vaccine at 0.6 or the standard 0.26, and there was a concomitant group of 16 to 26-year-old women that were enrolled, and this allowed 2 different comparisons. The 2-dose group at 9 to 13 compared to the women as well as the same age group that received a 3-dose schedule. These are the results from this study. The main comparison was for the 2-dose schedule in the girls compared to the 3-dose schedule in the women, and you can see here that non-inferiority criteria were met for all types, and in fact the antibody response was higher in the girls who received 2 doses with geometric mean titer ratios greater than 1. And this was the main endpoint for the trial. In the direct comparison of the 2 and 3-dose schedule in the 9 to 13-year-olds, you can see that the geometric mean titers were less than 1, meaning the titers were lower in the 2-dose group than the 3-dose group, and non-inferiority was lost for HPV 18 by 24 months and for HPV 6 by 36 months. These graphs show geometric mean titers through 36 months for this trial for HPV 16 and 18. And you can see that actually, even though there were differences, the antibodies follow the same kinetics in all schedules. Antibody reaches a peak at 1 month after the last dose, then declines and reaches a plateau. And for both the 2 and 3-dose girls, shown in red and black, they maintained plateau levels that were higher than those in the young adult women, shown in green. Last, I want to mention the randomized trial of alternative 3-dose schedules for quadrivalent vaccine. This study was conducted in Vietnam. 11 to 13-year-old girls were randomized to receive the standard regimen 0.26 or extended intervals 0.39, 0.612, and 0.1224. At 1 month after the last dose for the 0.39 and the 0.612 schedule, non-inferiority criteria were met for all types. And in a follow-up study at month 29 to 32, non-inferiority criteria were met for all of the schedules for all of the types. Now, in this trial, a serology sample was obtained both pre and post-dose 3, and the pre-dose 3 can be considered a post-dose 2 sample. And this slide shows the geometric mean titers after dose 2 for the 4 schedules, and you can see there was a trend for higher antibody levels before the 3rd dose with increasing intervals between dose 1 and dose 2, ranging from 657 in the standard schedule to 1581 in the 0.1224 schedule. And of note, because the schedules were different, actually the time between vaccination and blood draw after the 2nd dose also varied. For example, in the 0.2 6-month schedule, the time between vaccination and blood draw post-dose 2 is 4 months, while in the other schedules they were longer. And in spite of this, we still see the higher antibody titer with a longer interval between dose 1 and 2. Now I'd like to review the effectiveness data that are available from post-licensure monitoring studies. There are now data from post-licensure monitoring studies, and population impact on some early outcomes has been demonstrated in countries with high as well as though with moderate or low coverage. I'm not going to go over these, but I've just listed the variety of countries that have shown population level impact of HPV vaccine early in the vaccination program. What I would like to review are those studies that, vaccine effectiveness studies that have tried or looked at vaccine effectiveness by number of doses. And shown on this slide are the 4 studies that do provide some information on vaccine effectiveness by number of doses. There are 3 for quadrivalent and 1 for bivalent. The first 3 were data linkages studies that use registry data available in those countries. And the last was a serial cross-sectional study of HPV prevalence. 2 evaluated cervical abnormalities, 1 condyloma, and 1 type-specific prevalence. And I should mention that there are a variety of other vaccine effectiveness studies ongoing, including one in the US looking at efficacy against condyloma. Now, there are a variety of challenges and limitations to these studies that I want to mention before I present the data. First of all, early in the programs, these effectiveness studies are evaluating outcomes that are in the catch-up population, not those that were vaccinated in the target age group, so many people were infected before vaccination. Second, there are differences between 2 and 3 dose recipients. And third, that the evaluations do not examine the 0-6-month 2-dose schedule, but rather people who were on the traditional schedule may not have completed, that did not complete their 3rd dose. So I'll go through these briefly. This slide shows data from Australia. The outcome was cervical abnormalities. And they looked at both CIN3AS and CIN2. And what you can see, I'm not going to go over this whole slide, but the hazard ratio for those who received 3 doses for CIN-AIN is 0.53, and those who received 2 doses was 0.87, which was not significantly different from the unvaccinated. And for CIN2, the hazard ratio is 0.70 for 3 doses and 0.99 for those who received only 2 doses. Now, the authors point out the many limitations of this study. Even though they controlled for the differences, there could be some residual confounding, and they point out that women who received 1 or 2 doses were younger at age at first screening, indicating earlier sexual debut, older at vaccination, and had lower socioeconomic status. This is the second study from Australia. It had a different study design, but also looked at cervical abnormalities. And here I've presented results for the entire age range that they looked at, 11 to 27-year-olds, as well as restricted to the 15 to 18-year-olds. And for the entire age range, there was also a difference here found between 2 and 3 dose vaccinees, with the adjusted odds ratio being 0.54 for 3 doses and 0.79 for 2 doses, although that was significant. And for the 15 to 18-year-olds, the adjusted odds ratio is 0.43, showing higher efficacy in this age group than in the extended age group, and also that for 2 doses, 0.77. Again, the same limitations here apply that I explained for the other study in Australia. Now, this study of the quadrivalent vaccine effectiveness for prevention of condyloma in Sweden was an open cohort of all females aged 10 to 24 living in Sweden, and it was a large study using population-based registers. Over 1 million women were included, and over 20,000 cases of genital warts. And incidence was significantly reduced for 1, 2, and 3 dose vaccinees compared to those unvaccinated. But the incidence ratio differed in their primary analysis, with the incidence ratio for 3 doses being 0.18 and for 2 doses being 0.29. And this study did clearly demonstrate impact from less than a complete 3-dose series, but there were differences by number of doses. The authors point out that the main analysis was done with an interval of 3 months between vaccination and case counting. With a longer period between vaccination and case counting, more genital warts can be excluded that might be due to exposure prior to vaccination. And with a period of 5 months instead of 3 months, a statistically significant difference in the risk of condyloma was no longer observed between 2 and 3 dose vaccinees. I'm sorry, that should have been up when I just reviewed that. Okay, the last effectiveness was of the bivalent vaccine. It was a cross-sectional study of women 20 to 21 years of age presenting for cervical cancer screening, and they looked at HPV prevalence, and the data were linked to immunization registries, and here you see that the adjusted odds ratios, again, was difference for 2 and 3 dose vaccines, 0.43 for 3 doses and 0.68 for 2 doses. So in summary, this is just a summary of the immunogenicity, efficacy, and effectiveness data. The geometric for both vaccines, the geometric mean titers were non-inferior after 2 doses given 6 months apart in young adolescent girls compared to 3 doses in the routine schedule in women 15 to 26 years of age. Geometric mean titers were lower but non-inferior after 2 doses compared to 3 doses in adolescents of the same age group 1 month after the last dose. Some quadrivalent vaccines did lose non-inferiority for 2 types at later time points. And for both vaccines, geometric mean titers are higher with longer intervals between doses for a 2-dose schedule. For efficacy, there are 2 small post hoc analyses for the bivalent vaccine that found high efficacy, and for the quadrivalent vaccine, there may be more data available in the future from the study in India. For effectiveness, there are 4 post-licensure effectiveness evaluations that have looked at number of doses, 3 for the quadrivalent, 1 for the bivalent, and these did find lower effectiveness for 2 versus 3 doses. However, there are really many limitations and challenges with post-licensure effectiveness evaluations at this point in the vaccination programs, and I have reviewed those already. Now, there are a variety of remaining questions. I've just listed one here. I think the main question really is, will there be differences in duration of protection if there is equal initial efficacy? And we know that some longer follow-up will be available from some studies. Modeling efforts have looked at this, and we have some information or some information to inform us from those modeling studies. And 2 studies suggest that if 2-dose schedules protect for at least 20 years and the benefits of the 3rd dose are small, if they protect for just 10 years, then the 3rd dose may prevent as many cancers as the first 2 doses. And we intend to look at this question more in depth for the U.S. For later presentation to ACIP. I mentioned earlier that SAGE has recommended a 2-dose schedule for HPV vaccination, but before that time, several countries had changed to 2-dose schedules or to the so-called extended 3-dose schedule. The purpose of the extended 3-dose schedules was to allow implementation of the first 2 doses on a schedule that would essentially be a 2-dose schedule for the first 2 doses if it was decided later that the 3rd dose was not needed. So it was a 0-6, 60-month schedule. And these are the countries, just some of them, there are more, but I just wanted to highlight these. Quebec was the first jurisdiction to use this. They implemented the extended 3-dose schedule in 2008, and they changed to a 2-dose schedule last summer. British Columbia changed from a quadrivalent 3-dose schedule to the extended 3-dose schedule in 2010. They're reviewing their recommendations now. Mexico has been using extended 3-dose schedule since their national program in 2012. I know there's people from Mexico here, and they can update this if there's more information. Switzerland changed from a 3-dose to a 2-dose schedule for 11 to 14-year-olds in 2012. And England recently announced that they will change from a quadrivalent 3-dose schedule to a quadrivalent 2-dose schedule for their school-based program in the fall of this year. And again, there's many other 2-dose schedules. This is just an example of some of them that we're aware of. Just coming back to the U.S., which I mentioned earlier, there's been no submission of data to FDA for the bivalent. There are no plans to submit data for the quadrivalent because they're focusing on the investigational 9-valent trial program. And there's no data on 2 doses in the BLA under consideration, but a trial has been initiated by the manufacturer. This is some information from the trial, which is registered on clinicaltrials.gov. It's a immunogenicity study. It started in December 2013, and according to the website, the last visit of the last person enrolled will be in July 2015. And we assume it will take several months to analyze the data, and we would have data available to look at in the fall of 2015. There are 5 arms in this trial. There are 3 experimental groups. There's a 2-dose group at 0-6 months in 9 to 14-year-old girls, a 2-dose group at 0-6 months in 9 to 14-year-old boys, 2 doses at 0-12 months in 9 to 14-year-old boys and girls, and then there are 2 control groups, the 3-dose group in the 9 to 14-year-olds and the 3-dose group in the 15 to 26-year-old women. Now, I want to mention a little bit about the plans of the work group. Our plans had been to review and consider, and still are, to review and consider the 9-valent vaccine as a 3-dose schedule, and to consider 2-dose schedules when data from the 2 versus 3-dose trial of 9-valent vaccine are available. However, we have had other discussions about other options. And the main one, of course, is to consider a 2-dose schedule now for bivalent, quadrivalent vaccine in 9 to 14-year-olds. I want to emphasize that for older individuals, a 3-dose schedule would still be recommended. Because of the intersection of consideration of the 9-valent and 2-dose, it's a bit more complicated. And one of the things we discuss is if a 2-dose schedule is recommended, what would be the options when the 9-valent vaccine is licensed? These include waiting until there are data for a 2-dose schedule before considering making any recommendations for the 9-valent, recommending the 9-valent as a 3-dose schedule, so going back to a 3-dose schedule, or recommending the 9-valent as a 2-dose schedule with no data. And most of the work group members felt we should continue to review and consider the 9-valent as a 3-dose schedule and to consider 2-dose schedules when data from the 2 versus 3-dose trial are available, but there were definitely a minority opinion. Now, I just want to end this talk reviewing data on coverage in the U.S. I know all ACIP members are very familiar with this graph, which shows national coverage levels among adolescents from 2006 to 12 for all of our adolescent vaccines, and showing in contrast to Tdap and meningococcal conjugate vaccine, only 54% of girls in 2012 received 1 or more doses of HPV vaccine, 33% had received all 3 doses, and in the last 3 years there's been very limited increase in coverage and no increase between 2011 and 12. Of course, one of the reasons for the interest in 2-dose schedules is because of the potential to facilitate implementation by simplifying schedules, although we have no data that simplified schedules themselves would increase vaccine initiation. And here what's graphed are coverage for at least 1 dose, at least 2 doses, and all 3 doses from 2007 to 2012 in females. And you can see all these have a similar trajectory. 54% received at least 1 dose, 43% at least 2, and 33% 3. And there's a similar drop-off between 1 and 2 and 2 and 3 doses. So just to extend the timeline that Dr. Bocchini showed at the beginning, this is our tentative schedule if we move forward with our current plan. In February, of course, we had the presentations already discussed. Today, we heard the clinical trial data and the policy questions to be addressed by grade. In October, we would have grade economic analyses, hear the immunogenicity data in males, and present recommendation options. February would be the estimated earliest possible vote on the 9-valent, and October 15 would be the earliest potential data that we would hear on the 9-valent 2-dose trial that would be available. I want to thank everyone who's helped me. I particularly want to thank Eileen Dunn, who will be leaving and moving to Thailand, so you won't be seeing her anymore at ACIP. Thank you.
Thank you very much. Open for questions and comment. Yes, Dr. Herman.
Have any of the countries that have switched to the 2-dose schedule signaled what they might do with the 9-valent vaccine?
I've had some informal discussions with people about that. I think everyone's grappling with the same issues.
Dr. Dushan.
Thank you for that comprehensive review. What was the reason that a 2-dose regulatory approval was sought in so many other countries and not here?
I think we can, I'd like to turn that over to the manufacturers if they want to address that.
Go ahead.
Is developing the products and contributing to the evidence base that informs policy decisions. So we engage with policymakers and regulators all around the world, and the level of interest in the two-dose regimen has varied from country to country, and in Europe there was significant interest in it, so we did provide the data that was used both by regulators and policymakers to lead to that discussion. That being said, as you've seen from the presentations by Dr. Markowitz and others, we have been involved in generating data that are relevant to the deliberations that you'll be carrying out. And, as we have focused on, as you've heard, the development of the 9-valent vaccine, which you can tell is a very complicated program in and of itself, and wanting to make sure that the transition from the quadrivalent vaccine to the 9-valent vaccine is as smooth and effective from both a medical and public health perspective as possible, we felt that in the U.S., our efforts were best focused on delivering the 9-valent vaccine with the strongest data set to support it. And in the meantime, we are generating, as you've heard, data on 2 doses of the 9-valent vaccine, generating data on the 2 doses of the quadrivalent vaccine beyond what's already available didn't seem like the best investment of resources or time.
Dr. Friedland?
Hello, Leonard Friedland from GSK. GSK has submitted and has approved now 2-dose Cervarix, our HPV vaccine, in over 71 countries, and it's recommended as part of the immunization recommendations in a number of countries to use the 2-dose in their programs. In the United States, the story with Cervarix is slightly different. Because of the low uptake of the use of Cervarix in the United States, we're devoting our efforts with a 2-dose Cervarix program in other countries around the world, particularly in developing countries where there's a greater opportunity for the use of the product, particularly around helping with adherence and compliance with the schedule. Thank you.
Dr. Curran?
So I was actually wondering what the 2-dose schedule is that Merck is studying for the 9-valent vaccine. Is it a 0-6-month schedule, or is it?
I think you mean this, it's on this slide here?
Okay.
Right, so it's both the 0.6 and the 0.9.
And a 0-12.
I'm sorry, 0.6 and 0.12.
Thank you.
Ms. Pellegrini?
While we've rightfully devoted a lot of attention to the 3-dose versus 2-dose, I'd just like to encourage the work group and the committee to pay equal attention to the intervals. And I'll put in a plug for families here. The current intervals of 0-6, right, are incredibly challenging for families, right? And if we could go to something like a 0-12-24 and align it with an annual teenager visit, I think the uptake would increase dramatically. Getting a teenager to the doctor 3 times in 6 months is almost impossible.
Other question or comment? A quick question for Dr. Markowitz, and maybe you covered this, but in terms of going for previously fully immunized individuals with 3 doses of HPV-4, are there considerations other than 3 doses of HPV-9 for catching up? And I'm thinking akin to when the switchover went from PCV 7 to 13. There was kind of a algorithm to use. Not everybody would get the full complement.
Yeah, I first of all, for PCV-13, my understanding is actually did that study. They looked at giving a single dose to people who had had a previous vaccination. We don't have those data. And so, we're in a different situation. And also the age group was different that they were considering. So we won't have data for that. And we will be looking at what data are available. And I mean, that would obviously be something that we will be grappling with, the issue of revaccination.
Any other comment? Dr. Schuchat.
Yeah, I want to congratulate the work group for covering such complex information and queuing this up for the committee. As Lori's, could you move back to your next steps for the ACIP slide? Lori laid out the timeline that she anticipates the work group bringing data back to the full committee. And this entire timeline refers to the 9-valent schedule. And I think it would be helpful to hear committee input into what else you want to consider about the intervals and 2-dose in advance of other formulations potentially being available. Because I think if one is heading towards a fewer dose schedule, changing the interval sooner is better than changing the interval later. So just giving some feedback so the work group knows what you'd like to see, I think would be helpful.
Dr. Kemp.
Yeah, along those lines, I mean, it's clear right now, there seems, the data is very strong that we should go to 0.6 or 0.12. The only problem is that when 9-valent comes along, are we willing to do that with the 9-valent without those data? And I mean, it's a little bit of an uncomfortable situation. It's almost as if the 9-valent were coming along, we would right now change the intervals. And yet everybody recognizes the problem with having to make pretty rapid adjustments in primary care. This is a very difficult thing to do. So I guess I'd like to hear from the other committee members whether there would be any chance of us going, sticking on that kind of a, essentially an off-label recommendation for that interval.
Anybody willing to take that on? When I have to reflect on Ms. Pellegrini's comments that in a primary care setting, we're doing atrociously in terms of getting HPV coverage out there. That's been shown now over, 8 years? We're doing poorly. And part of that is just dealing with adolescents in primary care, and they do not come in for a follow-up in a month or 2 months. And as a parent of a 2-dose son right now, who was immunized on a 0-12 month, I can attest to how difficult it is to get somebody in. So I'm guilty as charged. But really looking at the realities of practice here, I think is incredibly important in terms of implementing a very good vaccine. Dr. Rengold.
Well, I guess reflecting back on the discussion we had about going from 4 doses to 3 for pneumococcal conjugate vaccine and the standard of evidence that the committee seemed to want, I'm dubious that I'm not quite sure we would have any more convincing evidence about fewer cases of cervical cancer down the line based on going from a 3-dose to a 2-dose schedule for this than we had for going from a 4-dose schedule to a 3-dose schedule for pneumococcal conjugate vaccine. I think the data will be probably no better. So I'm not convinced that, based on that discussion, that the committee is ready to do that.
Dr. Bennett.
Well, I hate to say this coming from me, but I have to say that I think the data are better here because there are actually comparative trials, and we don't have that for PCV. We have 2-dose versus, or 3-dose versus nothing. We have 4-dose versus nothing, but we don't have 3 versus 4. And there are some 2 versus 3 data here.
That's true for immunogenicity, but if you look at the data with clinical outcomes, the relative risks for the 2-dose schedule are not very impressive, and they're certainly, they don't look comparable to the 3-dose schedule.
Dr. Kemp.
However, those were all 0.2, right? Am I right about that? That did not compare 0.6 or 0.12, so we really have no data on that.
I wanted to show the efficacy data because I think there's this feeling that we will have a lot of efficacy data to possibly inform, but I think that the efficacy data will be very difficult, not just because of the schedule, but because of the catch-up population, which was done, and the differences between 2 and 3-dose vaccinees. So It's interesting to see the data. I'm not sure how informative it is for our deliberations.
I think I just have to toss out the other factor is, though, I do have a screening test in my clinical practice for cervical cancer, and I do not for invasive pneumococcal disease. Dr. Bikini.
I was just going to say, I think the opportunity for considering a two-dose schedule would be very different if we weren't looking at a 9-valent vaccine that looks like it's going to be licensed within the next, 6 months. So that would create a situation where we would be making multiple changes in immunization recommendations within a very short period of time, which I think could make it very difficult for the primary care practitioner to follow those recommendations or trust them as they evolve.
Dr. Shukat.
This might have gone by too fast for me, but could you remind us, Lori, what type of licensure we're expecting for application we're expecting for males for the 9-valent in terms of age groups and so forth? Because there was a discussion of off-label intervals and so forth.
So, and Dr. Dunn reviewed this. So the first licensure, which we're expecting at the end of this year, will be for females 9 to 26 and for males 9 to 15. The data on the 16 to 26-year-olds or the 15 to 26-year-olds we will have in October. So we will have those data. We'll be able to see those data, even though they will not have been submitted yet to FDA. So the feeling of the work group it would be very hard programmatically to have the quadrivalent vaccine recommended for the older males, but the 9-valent vaccine recommended for females 9 to 26 and males 9 to 15. So the proposal is to move forward with the recommendation for 9 to 26, for the full age group of females and males, even though it would be technically off-label for the 15 to 26-year-old males. But we will have data that we'll be able to see before that time.
And Dr. Pickering.
Yeah, Dr. Luxemburg said that we would have two-dose data at the end of next year. What type of data will these be? And secondly, are the antibody levels and kinetics of the 5 strains that are in the new vaccine similar to 16 and 18?
So to the first question, the data will be immunobridging data, comparison of immunogenicity at the peak, which means 1 month after the last dose, be it the third or the second dose. In terms of kinetics, we've looked at kinetics after 3 doses, in the ongoing studies, and the kinetics is similar between the additional types and the original types, which means peak 1 month after, decrease by approximately 70% at 12 months, and by approximately 80 to 90% later on, reaching a plateau.
Any further questions or discussion? If not, I think we will limit things to about a 10-minute break.